The topic of this presentation is features of biogenic nanoparticle formation in agarose gels and their effect on cell growth during bulk cultivation. Our research is related to bioprinting industry, and bioprinting in its essence is a task of composing cells in a particular order and ensure their proper growth so they can form a biological tissue or an organ. For this purpose, cells are immersed in special materials like gels, and combination of cells and such medium is called bioink. After cells are composed in a particular order using a bioprinter, a new challenge arises: how to ensure cells' proper growth so they can form a tissue. Proper conditions are necessary for cells' development, such as nutrients and oxygen. Also, products of cells' metabolism must be removed. But what medium can be used for storing cells? Hydrogels can be considered as such material. It has certain advantages, such as technological suitability for bioprinting, sufficient mechanical strength, high internal mass conductivity, biological compatibility, bioresorbability, and non-toxicity of decomposition products. In practice, hydrogels can be considered one of the most promising materials for bioprinting applications. What is a gel? A gel basically is a dispersed system of high molecular structure filled with liquid. Gels can be characterized by fluidity under certain conditions, elasticity, thixotropy, mass conductivity. Also, gels are non-stationary and non-uniform systems. What is the best way to study how nutrients can be delivered to cells through gel? Studying real-life systems, including stem cells and blood substitutes, require a lot of resources and time. Modeling systems can be used instead for performing quick and reliable experiments. Such model systems include yeast cells, gels like agarose, and simple nutrients like broth. How we can observe biogenic nanoparticles growth? The idea is that metabolically active cells are able to produce nanoparticles of reduced silver from a solution of silver salt in a liquid environment with cells for a limited amount of time. Here on the left picture you can see a cell surrounded by silver nanoparticles of different sizes and shapes, which are depicted on the right picture in a large scale. The size of biogenic nanoparticles depends on the regenerative capacity of the biological objects under this study. Dynamics of silver nanoparticle concentration growth in liquid was studied. It was found that over the time absorption of light increases at wavelengths typical for silver nanoparticles, which makes possible to conclude that the concentration of nanoparticles increase over the time. Diffusion properties of gels were studied using colored nutrients in order to visually observe the diffusion process. A 4-layer gel sample, which is depicted on the right, was composed with alternating layers containing cells and gel areas without cells. In such way, cells can be isolated from each other in one gel sample. For example, it can be useful if different groups of cells must form different types of tissues. Also, remote optical methods are convenient for studying cells in gels because there is no mechanical intervention in gel samples. Here, a scheme of optical experimental setup is presented with 2 working areas, camera, spectrometer, and a laptop for data analysis. 2 types of agarose gel with different levels of electroendosmosis were used. The first type is agarose that has a relatively high level of electroendosmosis, which is named as A0. It can be a chemical reducing agent of silver salt cations and a source of silver nanoparticles formation. The second type of agarose, which is named A1, which has a low level of electroendosmosis. It has no potential for chemical reduction of the introduced silver cations and is not able to form silver nanoparticles independently without the presence of living yeast cells in the gel. Having been prepared for both types of agarose, gels demonstrated identical growth of yeast cells, and corresponding spectra for both types of agarose gels are presented on the picture on the right. Transmission spectra for hydrogels with an agarose concentration of 0.4% by weight, prepared from agarose of type A1 at silver nitrate concentration of 5 micrograms per milliliter, were measured after 2 hours after preparation, after a day, and after 7 days. Obtained data demonstrates that an increase in the concentration of nanoparticles corresponds to a decrease in the light permeability at a wavelength of 400 nanometers, which correspond to wavelengths typical for nanoparticles, and for yeast cells at 600 nanometers. As it's shown on the picture, the lower the light permeability at 600 nanometers, the greater the number of yeast cells in the gel. Accordingly, the lower the light permeability at 400 nanometers, the greater the concentration of biogenic nanoparticles in the gel, as it's depicted on the picture on the left. Based on above-mentioned results, the following conclusions can be made. Adding silver nitrate concentrated below 5 microgram per milliliter can be used to determine the cell's state in the gel volume. Yeast cells, while growing and dividing, are able to restore silver cations and form nanoparticles. It is shown that for the purpose of determining the cell's functional state by this method, it's necessary to use agarose with a low level of electroendosmosis, and such agaroses themselves don't lead to the nanoparticle formation, and all the resulting nanoparticles are the product of the interaction between the silver nitrate salt and living cells. The ability of immobilized yeast cells to overcome a thin layer of gel containing recovered silver nanoparticles was evaluated using the technique of multi-layer gel structures. Cells germination due to division through a gel layer was not detected. This method can be used to prevent spontaneous cells migration in undesirable directions inside gel. Thank you for your attention.